So, a very warm welcome. As I said, this is our Both Sides Now art science event as part of Stobfest. So, great to see so many people here. If you're joining us for the Teams meeting, do make sure that your mic is on mute and your cameras, ideally for the audience, will be off so that we can see more of our fantastic presenters as well. So we're joined today by some fantastic people. So you can see we have Mattie, Pawlowic and Susan Wylie, who are scientists, and Janice Aiken and Emily Fong, who are artists. And we also have Ailsa Mackintosh, who is keeping us live and on the air. But if anything goes wrong, it's my fault. They're all going to tell you a little bit about themselves more as we get into it. But I want to introduce what we're talking about tonight, kind of what the event's all about. So this is a part of Stobfest, which is our local festival in Stobswell. We've run for the last several years in person, but this is our first time running online following the COVID pandemic. So we're very, very excited to be here. I'm here tonight both as a member of the Stobswell Forum, which runs Stobfest, and also as the public engagement manager for a part of Dundee University. And that part's called the Wellcome Centre for Anti-Infectives Research, which sounds quite a mouthful. What we do is we research new medicines to fight something called neglected tropical diseases, which are some very nasty diseases, often caused by parasites. They cause a lot of harm around the world. We do fight some other diseases as well, which you'll be hearing about in just a little while. And our missions include scientific research, so making those medicines. We have a program to train scientists from around the world, so we have brought them to Dundee to learn the art and science of what we do. And we also do a lot of public engagement. So working with people like yourselves who are here today to see what we do, to learn from you as well. But we also are incredibly keen on pairing our scientists up with artists. We've had a few fantastic projects in the past, and what we find is that our scientists learn a tremendous amount from thinking creatively with artists. Artists get to learn some really cool stuff working with our scientists. Everyone at home gets to see some fantastic new projects. There's just all this fantastic mutual benefit to everyone. So actually, we thought it'd be interesting tonight to start with a little bit of a Mentimeter quiz, a little challenge, which some of you may have done in these kinds of events before. So we're just going to ask you a little question, and then we'll give you a space to tell us your thoughts, ask us your questions, which we'll be keeping an eye on through the event. So Ailsa has seamlessly changed the screen over. Thank you very much, Ailsa. So what you'll want to do is reach for your phone, open another browser window, get another internet-ready device, and you're going to want to go to the website menti.com. Now that little box appears over at the moment. It will hopefully disappear in just a little second. So Menti is MENTI.com, and there will be a little code which hopefully we'll be able to see in a moment. That's the wonders of working on the internet.
I can confirm the code is 98183014. Oh,
that's brilliant.
The box is gone now.
That's what appeared on the screen now. So menti.com, MENTI, and your code is 98183014. And if you want to just take a little moment, we're really curious to see you. How strongly do you see yourself as each of these categories of person, which I'm sure we'll find out through the event, are entirely artificial categories. But yeah, great. We can see people voting already. Okay, we started very strongly at one end. A little bit of a balance going on there. This also helps us gauge how many folk are here with devices that are working tonight, which is always good. Okay, so we're getting at the different shapes are interesting. There are obviously a few people who've very strongly identified as not sciencey, and a few have identified very strongly, and it looks like there's a few more people in the middle who are kind of middling on artsy. Okay, good stuff. Good stuff. We'll just leave that for another little second, see if anyone else is going to join in and vote. And what we'll do in just a moment is we'll move that slide on to the next one. There we go, and again, extra little voter in there. Yeah, great, cool. Let's leave that with that for now. And we're going to leave the next slide. The slide will stay there on the Mentimeter, and you'll be able to put your questions in for us. We'll see them coming through. We'll do our best to get through as many of them as you can. So if you think of something while someone's speaking, pop it in there. We'll try and see if we can get those in there. We'll look forward to answering as many as we can. Can't promise we'll get to all of them. Time always flies by with these things. Okay, so we're going to hand back over now to our expert speakers, who are people we're really here to hear from tonight. So we're going to start with Susan Wylie, Susan, I'll let you introduce yourself actually, because you will do it much better than I will. So Ailsa, if we can pop back off the screen sharing, and this is doubtless when the internet crashes on us. There we go. So everyone at home, please give a very warm virtual round of applause to Susan as we give her the stage. Susan, take it away.
Hi there, everybody. Can you see my slides okay?
Just about to come through. It's thinking
Okay.
about it. There we go.
Great. Hang on. It looks a bit odd. Okay. Trying to get it to share.
That's what.
Anyway, I will just carry on. Okay, so my name is Susan Wylie. I work in a group called the Mode of Action Group at the University of Dundee, and I've been in Dundee now for almost 20 years, which is crazy to me, but during that time I've really developed a fascination with the parasites, or the little bugs that cause these tropical diseases that Ali was talking about. And these are some of the diseases that we are focused on, and some of the parasites that cause these diseases that are that you can see here on this slide. Some of these names will be familiar to you. I'm pretty sure most of you will recognise malaria, or heard of malaria before, and it's actually the world's biggest parasitic killer, and kills still today kills almost 500,000 people every year. So it's a phenomenally dangerous parasite. And I also work on some less recognised parasites, one called leishmania that causes a collection of diseases called leishmaniasis. You can see here in the far left hand side, and that causes approximately 40,000 deaths a year, and is a real big problem in areas of Southeast Asia and also in South America, and probably is not a disease that you are particularly familiar with. But one thing that's really, well, a couple of things that are really common amongst all of the parasites and all the tropical diseases that we work on here in Dundee, the fact is that these diseases predominantly affect some of the poorest populations in the world, so unduly affect developing countries in the world, and so this is really has a devastating effect on their economies and also on their populations. And the other thing that's really common about these diseases is that we don't have effective drugs or therapies to treat these diseases, and that's really what we focus on in Dundee, trying to develop effective therapies in order to better treat these neglected tropical diseases. So I wanted to really just show you this slide to give you an idea of what specifically happens in my lab. So we work really closely with chemists within the University of Dundee, and I don't know if that's my surreptitious mind that I have these chemists standing there and they look like children, because I've been in Dundee for 20 years. Sometimes I think they are like children. They're 5 years old, but they're not really. So the chemists work really hard in Dundee to develop chemicals that we can then use to try and see if they may kill the parasites that cause the diseases that I've talked about already. So we take the chemicals and expose these parasites to these chemicals, and basically we're asking the question, can these chemicals kill the parasites that cause neglected tropical diseases? And that is what we would call the start point for discovering a potential new drug. So, but the point is that we don't then know how these chemicals are effectively killing the parasites that we're working with. And we need to know that information in order to develop these chemicals into effective drugs for the future. We need to understand how these chemicals might actually work. What do they interact with within the parasites that the parasite needs to survive? And can we improve that interaction so that the chemicals can work more efficiently in killing the parasites? So that's why we've got this little Clouseau character there. That is how I think of my group. We are the Inspector Clouseaus of the drug discovery world, and we try to understand how these chemicals are killing the parasites. And we use a number of different methods in order to do that. Some fairly cool genetics methods that we look at the DNA of the parasites. That's what encodes the proteins. these are all big words that I don't really need to go into too much detail about, but basically we try to understand how these chemicals are killing the parasites, so we can feed this information back to these childlike chemists in the University of Dundee, and with this knowledge we ask, can we make these chemicals better, even better at killing the parasites, even more efficient? Ultimately, this is an iterative process, so this goes on forever and ever and ever. And then ultimately, we have we may develop a compound or a chemical that may become an effective drug of the future. And obviously, this is our ultimate goal: is to produce a drug of the future that in the future that will be able to help people suffering from these devastating tropical diseases such as malaria. So this is really in a nutshell how where my group fits into this drug discovery process. So I think that in our chats with Emily and Janice, I think that we've tried to convey the fact that we love our parasites. We think the parasites that we invest all of this time to work with are really beautiful to us. They are dangerous parasites, and they can cause some devastating effects, but I think everybody really does love their parasites, and we nurture them right up until the point that we want to kill them with the chemicals that we want to develop into future drugs. So when I was chatting to Emily, I wanted to show her this picture in particular, and this is what you'd call a transmission electron micrograph. So basically what it is we take the parasite and we embed it into a resin, so basically like maybe a jelly or a harder version of jelly, and then we take cross sections of that parasite. So basically it's basically like taking the bonnet off a car and looking at the engine. That's what we're doing here. We're taking the bonnet off of the parasite and looking at what's going on inside, and we can take these beautiful images. And you can see that in the top, the purple image here at the top, that's the parasite in its entirety. This is Leishmania donovani that causes leishmaniasis that I talked about, and the image that we're looking at is a cross section of that parasite. And you can see on the left-hand side, this is a cross-section of the parasite. You can see it's got a membrane around the outside. We've got on the top left-hand corner, you can see these sort of they almost look like transections of straws. These are what we call the flagella. So they are what allows the parasite to swim around and be motile through its life cycle. And so that's healthy. On the left-hand side, we have a healthy parasite. You can see the odd little balloon floating around in there. We call these vesicles, and that's just things moving around the cell, and the parasite is fairly happy at that point. But when we add one of these chemicals that I talked about that we want to kill the parasite, you can see what happens to that parasite on the right-hand side. And you can see that it really looks like a bigger balloon. It looks a bit fuzzy around the outside because it's losing its structure around the outside, and you can see that it's filled with these little balloons in here, these little vesicles. They're accumulating, and it just looks less organized, I think, than the one on the left-hand side. And you can see that something fairly dramatic has happened. When I was chatting to Emily, what I was able to tell her was that the chemical that this parasite is treated with basically inhibits a motor within the parasite that is responsible for recycling of the waste products of the parasite. So it's called the proteasome, and basically it munches up all it's a bit like a compactor. It munches up all the waste of the parasite as it's going about its daily business. And so when we treated this, we added this compound, this chemical, this process is inhibited. And I said it's effectively like your recycling centre going on strike. If our bin men don't come round, you know that it doesn't take very long before the rubbish starts to accumulate on the streets, and we have bin bags everywhere, and it's a disaster, and everybody's super unhappy. And this is exactly what's happened in this parasite that's been treated with this chemical, because we've inhibited their recycling centres. The bin guys in this parasite have gone on strike, and it's chaos from that point. So this is like, I really love this image because it's beautiful in itself. Just looking at the inside of a parasite, I think that's phenomenal. But you can really see, from my perspective, a drug, a compound, and a future drug. Actually, this drug is actually going to go into the clinic and potentially be a future treatment for this disease. You can see really the impact that has within the parasite, and you can see the devastation that it can cause. And this is really fascinating to me, and I think it's actually a really fantastic image. And I should at this point thank Sonia Monish from a former member of the lab who painstakingly made these images, and that I was able to pass on to Emily to try and inspire her artwork. And like I said, I think we all believe that our parasites are beautiful and aesthetically pleasing to us. We know what a good looking parasite is, and it makes us very, very happy. And it's really nice to be able to chat to the scientists, and they can see the beauty in these organisms as well, despite the fact that they're also can be devastatingly dangerous to certain populations. But that's kind of the balance with being a scientist, I think. So I'll stop there, and I don't know if anybody wants to ask any questions or.
Brilliant, thank you so much. We've actually had a couple of questions come in already, but in the interest of time, I think we're going to hand straight on over to Emily next.
Hang on, I might have to, because I'm going to share Emily's presentation. So
do a little bit of screen swapping. Emily can get herself online.
Okay. Hi, Emily.
Emily, you can unmute yourself. Eternal battle.
Hi there. That's better.
Right.
Excellent. So Susan's going to, I believe, share my presentation if that works for you, Susan. But just to briefly introduce myself, my name's Emily and my pronouns are she, her. And I'm of both Caucasian and East Asian heritage. I have round glasses and dark shoulder length hair. And I'm really, really excited to be here. And a bit of background for those of you who haven't met me before. I'm an artist who's currently working as artist in residence with the Wellcome Centre for Anti-Infectives Research, and I have been doing so for the best part of a year now, which has just been totally delightful and a really interesting experience, especially during the pandemic. A fun fact is actually that I've met many of you, many of the scientists, but I've actually never met any of them in person, so I'm looking forward to doing that someday. So tonight I'm going to share a process that I've gone through after speaking to Susan about her parasites. Her two images that she's just shown you now, they're extremely beautiful, and really in themselves I find them totally delightful. So I really didn't want to look at them, I guess, from the point of view of drawing them as they are, just because I think as they are is totally wonderful, and Sonia's taken a lot of time to get those images made. So instead, I have made a painting called Parasite Soup. And in the process of talking to Susan, she has, I guess, shared with me how she actually got to those images themselves. And I've, I guess, from there pulled out a few of her, I'm calling them her modes of action, but really the modes of action are the modes that, the parasite is kind of undergoing. But for me, in my creative process, I'm taking her science as my creative start point, and I'm looking at those 4 modes. So if you can go to the next slide, please, Susan. So the 4 modes that I have pulled out, and please excuse me if these are ridiculously oversimplified, but I've got mode number 1 as resistance, mode number 2 is amplification, mode number 3 is fishing, and mode number 4 as soup. So for my painting, I decided to actually kind of mesh these mode 3 and 4. So I decided to go fishing, but also looking at the soup. So with this mode number 3, Susan spoke to me about how essentially, the best way to find a chemical start point is essentially to throw a bunch of random chemicals at the parasite, and if it kills it, then brilliant. So also what we're doing here in mode number 3, fishing, is we're mushing up the parasites, so grinding them up, making a big vat of soup. And then essentially we're going fishing with the chemicals. So we're seeing what is actually going to be pulled up when we put our chemical into this big vat of soup. And, you know, what comes out might be a goldfish or a shark, according to Susan. She finds it very exciting each time because she, you know, doesn't know what's going to happen. So I decided to take this idea of, you know, not knowing what's going to happen, but using these creative methods to start making a painting. So if we can go to the next slide. So we spoke about the recycling center of the parasite being what is really not working after the process of this drug presence, this drug pressure. And Susan said to me that essentially, you know, what she wrote in her paper about this research was that the parasites were drowning in their own waste, which I don't know about you, but I find that quite visually disgusting and delightful at the same time. It's a bit delicious. So I decided to grab a few of my my waste products that I collect. I'm a bit of a hoarder, as many artists and I hear some scientists are too. And I wanted to make these little structures, which are kind of like parasites, but not. So they're me going fishing, but I'm also looking at the I guess the aesthetics of the parasite. And these yellow things are my flagella. So they're the little tails that propel the parasites along. But also what they're doing is they're my mode of action for being able to play with my painting as I'm making it, essentially. So what I've done here is I've laid out my parasites and my fishing devices with their flagella in an organized way on a big piece of watercolor paper. And, you know, it's starting to look pretty fun at this stage, the process. And I always find if the process is starting to look fun, then, you know, the outcome has got a better chance of looking exciting. So if we can go to the next slide slowly. So what I'm doing here is I'm literally drowning my parasites in, you know, they're not drowning in their own waste, they're drowning in my soup as such. So I've taken a couple of colors of watercolor that I'm really interested in, and I've wet the paper. So it's, you know, it's all very soggy, and I'm using extremely thirsty watercolor paper here. So it's 600 GSM for those who have experience with watercolor, but essentially it's really, really thirsty. So I'm laying a lot of water on and a lot of color and really just watching to see what is happening. And as you can see here, if you're looking closely, the color is kind of attaching to the different compartments of the nets. So that was quite fun. So I was really just feeding these parasites with color and watching how that color moved around. And also, as different stages in the process, I was also twisting the flagella because I was thinking that actually if these parasites were moving, then they could actually move on the paper as well. And we might be able to see how the watercolor follows that movement. If we go to the next slide. So I left my parasites drowning in their own waste overnight because, as I said, the paper is really thirsty and it takes a long time to dry. And I didn't want to mess with the process of that drying time because the watercolor really is, it was doing something very, very interesting with these parasites and these nets. So there comes a point when you're painting, even if you've got a strategy or a plan that I guess is aligned with concept. You have to let the process just run. You have to run with the process itself. So what I decided to do was, because I quite liked where the different nets or the parasites were touching, I thought that was quite magic where the communication of these different shapes was happening. So I actually added a little bit of gold ink just at those points where the communication was happening. Because I think it's, you know, it's really interesting also when you're thinking about art and science overlay. It's that overlap that is usually really the exciting bit. And as you can see here, I got a bit of a delightful surprise when I woke up this morning. But the paper was still quite wet. So the next part of Susan's mode was actually looking at the thermal stability of the parasites and this binding site. And she was telling me that actually it's quite interesting because when there's a tight bond, there seems to be a really interesting reaction to heat. So I thought, okay, well, this matches my process. I'm going to get my hairdryer and speed up the process. So with my thermal action, I've got the hairdryer here, and you can see that's why the nets are starting to move over, because they're reacting to the heat. And this is the final image of Parasite Soup. So I've removed all of the nets, and obviously you can see that, you know, the color of the netting doesn't affect the actual composition in itself. But the movement of the parasites is, I'm actually quite delighted with this image. It's come out quite nicely. And yeah, the gold, I think, really starts to emphasize that movement. So there you go. There is my Parasite Soup.
Fantastic. Thank you so much, Emily. It's so fascinating to see it coming to life and the dimensions that it has grown. I'm aware of time, so we'll move on quickly, but we've got one question which I do want to bounce off you right now. So how have you found not actually being in residence with us? Has it affected your ability to understand our processes?
I think it's been absolutely fantastic, you know, and there's something quite unusual about working in this format. And as I've had quite a few conversations with many of the scientists, and said actually that this format of talking online and being able to have discussions in a private space is actually quite ideal for this kind of collaboration. So
it's almost a kind of different level of intimacy, isn't it? It's really fascinating. Yeah, great stuff.
So.
Great stuff.
I absolutely love it, Emily. That's the first time I've seen it, so I'm like delighted. That's absolutely gorgeous. Well done. That's, yeah.
Thank you so much.
And especially like the communication, because the parasites do in some ways talk to each other. They send signals backwards and forwards and communicate when they've got enough of something or not enough of something else. And yeah, so that communication and the gold representing the communication between the parasites, I think, is phenomenal. Thank you.
Great stuff.
Thank
okay,
you.
So we're going to keep pressing onwards. So next we're going to hand the floor over to Mattie, Pawlowic, who's going to introduce herself. So Mattie, again, a virtual round of applause and take it away.
Thanks, Allie. I'm Mattie. I'm a white lady with short brown hair and glasses, and my pronouns are she and her. I'm just going to tell you a little bit about what we do, and Janice has made some really beautiful paintings like Emily that are pretty visual, so I let her paintings, which are a little bit more literal, show you what our parasite looks like. So my group in the University of Dundee, we have about 6 or 7 people. I have 3 PhD students, 2 postdocs, and a research technician. And we study cryptosporidiums. So those are one of the parasites that Susan's group, we collaborate on together. Crypto causes diarrheal disease, which is never very fun to think or talk about. But it's really important because young kids around the world die of diarrhea at really high rates. And crypto causes really, really severe dehydration in these kids. And the children that we're talking about are mostly kids that are malnourished and in parts of the world where they don't have access to clean water. And cryptosporidium is spread through contaminated water. And that is something that my group is really interested in learning more about, how crypto can travel through water. And at the University of Dundee, we're also collaborating with Susan's group and the Drug Discovery Unit to make new medicines for crypto, because there's no medicines and there's no vaccine. And so for kids around the world who don't have access to clean water, crypto is a really big threat. And so we're working together to try to improve that. And then just on the personal level, before I was a scientist, when I was a kid, I was really into art. I was the kid who always got art supplies for Christmas and my birthday. So it's been really fun to talk with Emily and Janice and to revisit. I'm a really bad painter. Other kinds of art, I'm okay, but it's been really fun to think about art again and to see creativity from science and from art. So yeah, I think I'll turn it over to Janice now, or back to you, Allie, to turn it over to Janice.
Thanks so much, Mattie. And I think it's worth saying that there's really very little known about cryptosporidium in the world. The work that Mattie's doing in Dundee is world leading. That's one of the things the city does, so amazing stuff. So delighted to have you in Dundee, Mattie. So Janice, again, we'll hand on over to you. A virtual round of applause from everyone for Janice Aiken.
Yes. Just in order to introduce myself, yeah, I'm an old woman with long hair and glasses. I would like to just show you some pictures. I know it's a bit of a cop-out, but as an artist, I have to just work from the visual. So what I'm going to do is just quickly share my screen. Here we go. Is that okay?
That's working brilliantly.
Perfect. So my name's Janice Aiken. I'm a reader at Duncan & Jordanson in art and design, and I popped this in just to say I have an interest in representational painting, but I've also got an interest in really abstract, colourful, design-based work. Colour is my thing. I absolutely love colour. I'm not going to go into too much detail about that until I show you some colourful pictures. But I just wanted to say this is the sort of work I've done before in collaboration with scientists. The top one is the windows. I designed the window panels here at 9 Wells. Richard III, some of you will have seen that. I painted that guy's face. Which was a very big project, which was most bizarre, to be honest, having a sort of bust of Richard III sitting in my living room and painting his face. This is a piece of 3D, from a 3D animation that I made about the eukaryotic cell, which is kind of one of the basic forms of cell. This piece here is actually related to the work that the teams we've been looking at do. This was Professor Ian, a piece of work from Professor Ian Gilbert's lab, and this is a bronze model of a compound, a molecule from a compound that may have an effect on malaria. And I just popped that in because some of you might have seen the penguins, that I designed the sculpture of the penguins. So just to let you know who I am. And also, I just want to say one of my first glorious kind of ventures into looking at images of that are taken using various different microscopic techniques. It was a project using stem cells that was in collaboration with Dr. Mario Stavridis, and I've actually still got these 3 paintings on my wall behind me at the moment because I just love them. I love the image, the shapes, the colours. So this slide here is a selection of the images that Mattie and her team were able to share with me. And again, I completely understand what Susan said at the beginning of her talk. The shapes, you know, these are like beautiful drawings. They're like, you know, Leonardo da Vinci drawings or something. You know, the shapes and the way in which each object kind of, you can see how it's interacting with all the bits of the human body around it, or the body around it. I'm not sure that these are all human. And the different forms and the different stages of the Cryptosporidium organism. I just find them absolutely fascinating from a visual point of view. And I mean, I think that Emily's work is absolutely fantastic. It looks beautiful. And I love the way that she's taken concepts and translated them. My approach was a lot more literal, a lot more about almost like making a portrait of these objects. You know, these organisms, living organisms that have an incredibly interesting life cycle. From a perspective of a sociologist, they are horrific, but from the perspective of someone who's studying them, they're actually gorgeous. And, you know, I loved what Susan said about the fact that they love their organisms and they nurture them and cultivate them. So these are the images that I started off with, a selection of them. And these are some of the sketches. So this is work from my sketchbook. So you can see already that what I'm trying to do is take what these images are saying and kind of interpret them in a kind of colourful way, interpret them in a way that is almost abstracting them, but that still maintains a little bit. I mean, this one, for example, was taken from, specifically drawn from an object, from a, hang on. Is it? Have I got it there? It's the same organism as this. Sorry. So these are just some of the sketches that I did. And from all of this work, I've actually been experimenting. Now, these images are slightly more finished. These are painted with alcohol ink, which is really interesting because part of that process of making the image is about applying the ink to a sort of shiny surface, and it does things that you can't predict. So some of these shapes that are created by just dropping ink onto the surface actually quite resemble the sorts of processes that are happening within these organisms and the little organelles within them. So this is sort of at the moment the culmination of what I've been doing in terms of my art output. However, my final slide is this one, which is basically showing you some of the paintings that I've been doing that are much more about illustrating the different stages of the life cycle of the Cryptosporidium organism. So I'm going to hand back to Mattie so she can have a little bit of a comment on these images, and also to perhaps explain a little bit what they are images of.
Yeah, I'm happy to do that. I also want to say it's so fun to see color infused into these kind of images, because Susan showed, you know, this black and white photo, and also some of the photos that we sent Janice to get inspiration from. They're also collected in a way where we don't see color. We just see kind of shades of light and dark. So it's been great to see so much color brought to these parasites. Yeah, so Cryptosporidium, like other parasites, has a really complicated life cycle. It's spread through the water, not through an insect. And the life cycle that we find in the water is this painting number 3. So it's a really round form, and the parasites are these blue kind of snake-like shaped organisms inside, and there's 4 of them. So you can see those 4 dark spots. That is where the DNA is in each of the 4 parasites, and they're protected in this pink. I love this pink shell. So my group is studying what makes this shell so protective, because when the parasites are in water, you can bleach the water, which will kill everything else, but it won't kill Cryptosporidium. And so that's kind of the start and the end point of an infection with Cryptosporidium. And then all of these other paintings that Janice have made, these are crypto in context of the gut. So because it causes diarrhea, you know, you might guess that Cryptosporidium is found in the small intestine. And so the crypto grow in close connection with the cells that are in the intestine, and they multiply over time. So in painting 2, Janice is showing you how they go through this asexual replication where 1 becomes 2, becomes 4. And they also have lots of different compartments. So in painting number 1, this is a female. You might be surprised to know that all of the parasites that we work on, for the most part, although they are only 1 cell, they do have male and female forms. So they aren't like us, where they have an X and a Y chromosome. They have all the genes to be male or female, and they decide, every parasite decides if it's going to be male or female. And so number 1 is what a female looks like, and number 7 is a male. So you can see they have really kind of different biology, and the male also kind of has a little mustache. So that's quite fun. And you can see in 6, these are males kind of escaping and searching for a female. And in number 5, Janice has shown you the compartments within the cell, just like Susan and Emily were talking about these structures within the parasites. You can see more than just the DNA structure here. There are compartments that really are facing the pink cell that is the human cell. And those compartments are really important for the parasite being able to survive inside the human cell. And in 4, you can see there's parasites interacting with the human and also interacting with the contents of the gut. So yeah, I think these paintings are super, they bring a lot of energy to something that in black and white, you know, feels a little bit different. So yeah, I've really enjoyed seeing all the color and the energy from these, like the males bursting forth in 6. You can just feel it.
No, that's really, really great. And just to say that this is the start of a bigger project, I think, for us, in that these images that you can see just now aren't the entire life cycle. There are sort of in between stages that we haven't shown here yet, but I'm on it. They're on my easel as we speak, and I think that we are hoping that we can sort of do something with these that will allow us to create resources that will encourage people to understand the complexity, but also the importance of these neglected tropical diseases, because, you know, as everyone has said, these are incredibly important organisms if they are killing children, and you know, clean water is one of the basic human rights, and it's absolutely, I mean, I cannot tell you how much I admire the labs that are working on these diseases, because they will have real human impact. So I'll stop sharing now. And Ali, if you want to go for questions.
Fantastic. And actually, it's perhaps worth saying, I know we'd said that we would finish the event around about 1:45. It's been so interesting, and it's just about 1:45 now. So those of you listening at home, we're going to keep chatting, and you can listen in, you can take a break. We'll put the video up on YouTube anyway. So, but do keep with us, keep asking us questions, and I'm sure my panelists don't mind keeping chatting, because I can never. You know, it's always fascinating being in this group. So let's see. And yeah, we have had a question come in about what's happening with the work next. And that's one we'll be sending some emails out tomorrow after the event, and we'll got some links in there so you can find out more about us and see where the work's going. But yeah, we have lots of plans for as things continue reverting perhaps towards normal or some kind of normal. So let's see, which question should we start with? Let's start with a positive one for this one, Ella. So to scientists, do you find that working with artists benefits your research? And if so, in what way? Who wants to start with that one?
I'm happy to. I think it's great, because when you talk to scientists, and actually anyone that's not, because when you're a scientist, you're really immersed in the subject, and it's great to get, and maybe you hear the same kind of voices that talk about things again and again and again, and you don't get a completely different perspective. And then speaking to Emily, questions that she asked were not questions that a scientist would ask, but they were incredibly valid and made you just think of something, you know, in a completely different way. And they can relate the science to real life situations as well, and that helps us to communicate things better as well. And the fact that they can see the beauty in the things that we're doing is really incredibly, because you just think, as a scientist, quite often think you're very weird, thinking these things, oh, look how pretty, you know, scientists will show each other things and go, look how pretty this parasite is, this looks gorgeous. But then for someone to come and say that is absolutely beautiful, it's incredibly gratifying that you're not as weird as you thought you were. But Emily did that during our conversation, ask questions that really made you think about things in a completely different way. And I think that's really important to take, you know, comments and chat to people about science, to give you that different perspective, because, you know, that can flip everything on its head and make you think of something a completely different way.
Yeah, great answer. Great answer. Mattie, anything to add?
Yeah, I was going to say, actually, Janice asked some really great science questions that we don't know the answer to. And there are things that people in my group are also asking, you know, or sometimes we look at the photo and Janice is like, well, how many of these kinds of compartments are there? Well, we're trying to find out that answer, but we don't know. So I think sometimes the questions that artists can bring forward are really things that are super fascinating and really important to understand, and it just reminds you how little we know. So it's kind of nice to have affirmed that those big questions are things that other people are curious about too.
Yeah, because
I know that Mattie said that we work on these parasites are single celled, so they're not very, in the greater scheme of life, they're not very complex, but you can spend your entire career trying to understand one tiny aspect of this tiny cell. My granny used to say to me, have you not sorted that out by now? What are you doing? But yeah, these things are quite complicated, even though they're one cell. But yeah, my granny was very disappointed in me.
I'm sure she wasn't really. Okay, great answers. Thank you both. Okay, so I'll bounce one to the artists next. So I'm going to read this one out. So to the artists, much as you love scientists to work with, what scientist traits do you find the most and least attractive? Wait, I'm reading it verbatim
This could
here.
take a while.
Most attractive.
I can take that one. I mean,
Yeah.
I think the least attractive things about scientists are not about individuals. I think the individual scientists that I've met and worked with have always had the most joyful kind of enthusiasm for what they do. And Susan and Mattie are absolutely no exception. I mean, they absolutely love what they do, and they're really, really interesting to talk to. I think an unattractive scientist would be someone who doesn't feel that they can learn anything from others. I really genuinely have not come across that individual yet. Well, maybe a couple of people, but then I didn't work with them. But no, I mean, genuinely, I think it's really, really, it's a kind of almost a misunderstanding about what scientists do. You know, what they do is very, very creative. You know, that leap of faith that they take, the process that they go through is very similar to the process that we go through as artists. So I haven't really met the unattractive traits in scientists I've worked with. The most attractive things about them are their curiosity. A curious mind for me is just the most interesting person to engage with. Somebody who asks questions about the world around them, and you know, that for me is just, it's absolutely inspiring. I mean, honestly, the conversations we've been having, you can see probably from, I mean, all the bits of artwork that are all around me and the slides that I've shown, it's been a joy, you know, because there's this enthusiasm, and every time we speak, we energize each other. So there you go. There's a not particularly comprehensive answer. Emily.
It's an excellent answer, and I think really there's something that's so passionate about scientists. You know, everyone that I've met has such a love for what they're doing, and that's really like just super inspiring. I think it's an interesting question in the sense of what's unattractive about working with scientists, and I don't think that's. I think in the greater scheme of things, there's a mindset that, you know, there are silos between what people do. And that's not necessarily an unattractive thing that's generated by a scientist themselves or the science itself. It's just the thought that, well, you're doing the creative stuff, I'm doing the science stuff. Actually, they're kind of the same thing. It's thinking. And I think as soon as people understand that the thinking is really, it's free, you know, you don't have to be over here or over here. Then there's more ease between conversations between artists and scientists. But it's, yeah, I think that's a general wider mindset that's not necessarily the responsibility of the scientist, but it's something that needs to be changed slowly.
Yeah, cool, good stuff. Okay, great question come in here then. So who did it first? Did Susan decide to show the Parasite pictures to Emily, or did Emily specifically ask to see Parasite pictures, or did Emily just say, show me everything you have?
I was just thinking, what have I got that's cool to inspire Emily? I think that was it, and I love that picture, to be honest. I think that's of my career, I think that's my favorite picture, because it's kind of pretty, and it's also incredibly informative, and it ticks both boxes. For me it's really aesthetically pleasing, but also it told us something, and it was something important, and it demonstrated what was happening in that cell at that time, and that was worth 1,000 words. And yeah, so I don't know if it was, yeah, I was desperately thinking I need something really cool to show Emily.
Susan, she said to me, oh, my work isn't very visual, and then she pops up this beautiful image. I was like, yeah. No comment.
Actually, while we're here, Mattie and Janice, what was your kind of process? You know, was it similar? Was it different?
Well, Janice told me that she loved cells from the beginning. So, and I think Actually, since we've come out of lockdown last year, we've taken some of the best photos of the crypto that we have in the lab since then. And so something else that we do that was in one of Janice's sketches is we make them glow by turning them fluorescent colors. And so once I showed Janice the first fluorescent crypto, it was like over. So yeah.
Can I just say, we didn't have time today and I didn't have enough room in the presentation, but I have been also, in addition to these ones, I bought myself a UV light, and I've actually been doing paintings on black with UV paint to see how that'll be the next stage of
Yeah, that's
I want to see those.
Yeah,
Yeah.
so
They're like disco parasites.
Well, it really excited me, this idea, because it's not even so much about parasites, it's about how we see things, and it's about how, you know, what markers are you using in order to stain different parts of the cell. So yeah, I got really excited about that. But I think that the main thing, I mean, the first conversation we had, it was about, look, here's where I am, and Mattie could say, here's where I am. What can we show each other that will kind of inspire? And I think it's just been one of those iterative processes where you know, Mattie shows me something, sends me an image, I riff off that and do something interesting with it. And it actually genuinely during lockdown, it's been a bit of a lifeline for me, because having that connection and having these conversations is actually really good for the mental health. You know, it's inspiring each other and just having fun as well. It's really important.
But it becomes this lab-like process, doesn't it? Where you do run experiment, you see what the data is, and you keep going with it. They run in parallel beautifully.
That's what I really liked about Emily's image, because, you know, she incubated it overnight and she blasted it with some heat, and, you know, it's an experiment, and just like and just like, you know, science, you have no idea how it's going to come out. Sometimes it's not so good, and sometimes it just is amazing. So I think that's one of the things that people don't understand about science, and I'm sure this is true with art as well. Another parallel is that it can go really wrong. You know, it doesn't work all the time. You always get to see the perfect thing at the end. That's not always how it works. And I'm pretty sure in art, you know, you just try things. In science, you just try it. Is that going to work? Let's have a go. Okay, no, it was a disaster. Talk to no one about it and move on. So yeah, there's a lot of parallels.
And that's been something that's been really important for me during lockdown is I've been doing loads of experiments with different materials and things, and some of them are really successful and some of them are really just not very interesting. But the point is that you learn so much from the mistakes. Sorry, Emily, you were going to come in and say something.
Oh, no, I was just about to interrupt. I just said, I was thinking that, you know, it's quite a risky thing making art, you know, and actually I was thinking, well, the process is beautiful enough, you know, and we've had a lot of conversations actually about failed experiments and about how failure is important. So I thought, well, I'll use that mindset. If it's an absolute disaster, then we'll talk about failure. But yeah, I was quite hesitant to go down and check it, because I just thought, oh, this is either going to be lovely or really a hot mess. Like we mentioned, the parasite in it drowning in its own waste was a hot mess. I thought, well, if the painting is a hot mess, then we're also winning.
This is exactly what it's like when you come to you know, to the crux point of an experiment. You've got that mixture of dread and excitement. You're thinking, yeah, it could work or it could be a complete disaster. But it's always exciting.
Yeah, I have to say, when we started working with Emily, we weren't sure what the project would look like. And I said, Emily, please, could you kind of document and share your process? And Emily, I have to thank you for doing that, because I know it's not always comfortable to share the middle of an experiment. But I have absolutely loved seeing how this project has grown and grown, and all the offshoots that have come off from it. So it's been incredibly exciting. And this is another step in that. There's some great comments coming through as well, which I just want to share with you. So there's a comment: Parasite Soup is beautiful, especially with the story of how it was created. It brings it alive even more. Yes, couldn't agree more. Comment, Janice's paintings make me feel as though I'm looking at an alien world. Amazing colours. Thank you to the ex-biochemistry retrained as an illustrator for coming along. Great to see this. I suppose just for our last couple of minutes, the question was what enticed the scientists to want to work with the artists? But actually, I'm going to throw that out to all of you. What enticed you all to work together beyond Ali made you?
Who wants to go
I think
first?
maybe we're all envious. The scientists want to be artists and the artists want to be scientists. This is how I feel, is that it's really fun to play in these different kinds of ways. So, you know, it's like we get to be an artist just by chatting with artists and seeing kind of what they're doing. So I certainly would, you know, enjoy sometimes. I mean, the artwork I would create would be nowhere near, I think, as wonderful as the art that you guys have. But it's so fun to watch that happen.
Did you see how Emily and I both looked at you when you said that? There's absolutely no reason. We would both love to, once we can get together, once we can actually physically be in the same room, we would love to do some kind of session or something with you where you're actually making stuff, because actually, you know, it would be different work, wouldn't look the same, but it's still about that creative process. And, you know, that's what attracted me to working in this way. And I have worked in this way quite a few different times, is just because it's about sharing and working together. And I'm going to sound really gushy now, but there's just something lovely about a collaboration, particularly when you're bringing different things to the collaboration. You can almost always, if you can get a bit of chemistry between the collaborators, you can always get more out of the end result than you would if you were just sitting as an artist. I would be sitting in a little garret just drawing things, you know, nothing wrong with just drawing things, but I do really enjoy that collaborative process. So that's what attracted me to the whole thing.
Yeah, I agree with Mattie. It's completely an envy of the skills of the artist, and to just in a small way to be able to try and absorb some of that, and to be able to live in that world a little bit, because I'm not known for my artistic talents, that's for sure, as my art teachers will tell you back at school. But yeah, I think that it's I'm inspired by what they've been able to do and how they can interpret our work in such a beautiful and interesting way. So yeah, it's just an envy of their skills, really, that what's made me want to do this. So.
But you guys are saving people's lives, you know, and if we can even contribute a tiny bit to that, you know, to these ideas that you're having, and actually, you know, this is real world stuff. This is something incredibly important. So it's a joy to be part of it, you know, even if we can just chip away a tiny bit at that process. So yeah, probably it is a little bit of wanting to understand what each other does, but yeah, anyway, it was great fun, and I would really want to continue doing it.
And Emily, anything?
Well, I want to say that, you know, artists, we save lives too, you know, in a different way. But also, you know, I think, Susan, you said you're not known for your artwork, but yet, we put the word yet on the end of this. You know, I think it's something that everyone has the potential to really shift the way that you think and the way that you do things. And I love that, Mattie, you're talking about how, you know, you're excited about being part of the process of the art, you know, and that really is the case. It's not like Janice and I have gone off and made these things independently. We've made them because of your contributions, you know, and that's really exciting. I love that part. I love, just sharing that whole process is really, really exciting for me, but also just the shift in perspective. That's a huge one. I love that.
Absolutely.
Fantastic. Well, thank you all so much for taking part, for giving us your time, your wisdom, your creativity for the event. It's been fantastic. In terms of kind of Stobfest and Wellcome Centre, the Centre and the Forum have been working together now on this kind of public engagement stuff for a few years. And so it's really exciting to be able to bring this kind of project into Stobs Well and to have conversations with all of you, with everyone asking questions. So thank you everyone at home for asking those questions and giving us your comments. We really, really appreciate it. Ailsa, thank you as well for keeping us on the air and keeping the questions running as well. Much appreciated. Stobfest tomorrow night, we've got at Hame with Sheena Wellington. Do come along to that. Living legend, Sheena Wellington. Come on, all of you, you're very welcome. Tickets are on Eventbrite. We'll be sending an email out as well with a little survey to ask what you thought, and we'll share some links so you can keep following each of these fantastic people and see where the project goes. So thank you all very much for coming along. Have a fantastic rest of your night, and we'll hopefully see you again at more Stobfest and more Art, Science and Welcome Centre events too. Have a great evening, everyone. Thank you.
Thanks, guys.
Bye.
Cheers. Thanks.
Bye.
Stop recording.